Today I'll be talking on the spore staining. So this spore staining has three main objectives to why we are doing the spore staining. Number one is to detect for the presence of an endospore. Number two is to identify endospore producing bacteria, and third one to differentiate between the vegetative forms and the endospore. So the principle of this spore staining is primary stain malachite green is forced into the spore by steaming the bacterial emulsion. Malachite green is water soluble and has a low affinity for cellular material, so vegetative cells may be decolorized with water. Safranin is then applied to counter stain. Any cells will have been decolorized. At the end of the staining process, vegetative cells will be pink and endospores will be dark green. Spores may be located in the middle of the cell, at the end of the cell, or between the end and middle of the cell. Spore shape may also be of diagnostic use, and these spores may be spherical or elliptical. So in the spore stains, we're going to be using the malachite green, the decolorizing agent, and the counter stain. So let us see what does this do in the staining. So in malachite green, unlike most vegetative cells type that stain by common procedure, the free spore because of its impervious coat will not accept the primary stain easily. For further penetration, the application of the heat is required, and after the primary stain is applied and the smear is heated, both the vegetative cells and the spore stains will appear green in color. So that is what the malachite that is how the malachite green work. And then the decolorizing agent. Water once the spore accept the malachite green, it cannot be decolorized by tap water, which remove only the excess primary stain. So the spore remains green in color. On the other hand, the stain does not demonstrate a strong affinity for vegetative cells component, and the water removes it, and then the cell will be colorless. So that is how the decolorizing agent works. And now let us see the counter stain, the last one, which is safranin. So this contrasting red stain is used as the second reagent to color the decolorized vegetative cell, which was decolorized by the decolorizing agent. So this will absorb the counter stain and appear red in color. So the spore retain the green of the primary stain, that is the malachite green. So this is the principle of the spore staining. So let us see the requirement that we are going to be needing to perform this spore staining. So the first one you'll be needing is the malachite green, the safranin, and also the glass slide, then the inoculating loop, then also the Bunsen burner, and the specimen. So for the procedure, you have to cover the smear with a piece of absorbent paper or a tissue paper. Then place the slide over a staining rack that has a beaker or water bath of steaming water. Then flood the absorbent paper with malachite green and let it steam for 3 to 5 minutes. Then you have to remove the stained absorbent paper carefully and discard and allow it to cool for 1 to 2 minutes. Then gently rinse the slide with tap water by tilting the slide gently to allow the water to flow over the smear stain. This is because this is to remove the extra dye which is present on the slide on both sides, and also to remove extra dye staining any vegetative form in the heat fix smear. Now you have to add the counter stain safranin for 1 minute. Then rinse the slide with running tap water or on both side of the slide to remove the safranin reagent. Now you have to ensure that the bottom of the slide is dry before placing it on the stage of the microscope to view with the immersion oil. Now after this, so this is the procedure of the uh, spore staining. So after this procedure are done, you are going to observe in the microscope with the immersion oil. So after observing, you are going to get a result of you're going to get a result like this. So here in the given image, let me give the result of this. So the vegetative form will take up the pink or the red stain from safranin, while the endospore will stain green from the malachite green dye. Now let me interpret this result. So the vegetative form stain pink or red. This is because they take up the counter stain, that is safranin, while the endospores take up the green from the malachite green. This is because during smearing and heat fixing, the malachite green penetrates into the endospore with the help of the heat from the steam, and during the water rinse, the dye is not easily they did not easily get washed away, and for the vegetative form, the dye is easily washed away because of their fragile outer covering. So for this case, they take up the last stain, which is the counter stain. That is why they appear pink or red in color. So this is all about the spore staining.